Process design.
When dealing with the production of a recombinant protein, it's necessary to analyze the basic principles of the main expression systems that are available in the laboratory in order to design a proper procedure for the production, purification, and characterization of the recombinant protein. The recombinant gene is then introduced into an expression vector, which is transferred to an expression cell to induce the expression of the recombinant gene to produce the recombinant protein. Under some circumstances, the protein gets a properly fold conformation, which can be then used for the purpose of the project. However, in some cases, the protein is accumulated in the insoluble cell fraction as misfolded conformations, or the protein is not detected at all due to problems in the transcription or translation or proteolysis during the expression experiment. The recombinant gene needs to be redesigned using bioinformatic tools, and the process starts all over again in order to obtain the properly fold conformation of the protein. Inside the cytoplasm of the expressing cell, before inducing the expression of the heterologous gene, it has been seen the presence of a crowded cytoplasm in which the recombinant protein is going to be accumulated at the same time. Therefore, when producing a recombinant protein, the cell is under stress conditions, and this it's related then with the appearance of protein in the aggregated form or the proteolysis. This image it's common for eukaryotic and prokaryotic cells. So these problems can be encountered in both type of expression system. Provided that the cells need to cope with the properly fold conformation of the proteins, all cells have a protein quality control system that has been widely described for prokaryotes, as indicated in this slide. However, in eukaryotes, this system has been conserved. So the nascent polypeptides are combined with protein factors which lead the protein to a proper conformation, giving rise to this properly folded conformation or the native protein. However, in some cases, this protein is not able to obtain the final folded conformation, and then there are secondary conformations which can be then taken by other proteins in order to get this native conformation. If this is not the case, these secondary conformations can then be accumulated in the form of aggregates. There are some other factors, these aggregates, that are able to transform those aggregates into those secondary intermediates, which can be then be in contact with the chaperones in order to obtain the native conformation. However, in this process, it's also connected to proteolysis, to the main proteases of the cell and also minor proteases, which are then in the process of in the pathway of the recycling of the amino acids that are accumulated in the cell in the form of aggregates. So, in this context, the addition of a new heterologous gene then will saturate this protein quality control system, which has been evolutionarily selected to cope with the metabolical needs of the expression cells. So therefore, the objective of the recombinant protein production is to obtain the protein in the soluble cell fraction. In most cases, the protein, even though it's produced, it's proteolyzed due to this connection with proteolysis, or the accumulation of the protein in the insoluble cell fraction in the form of protein aggregates, which are known in prokaryotes as inclusion bodies or are known in eukaryotes as aggresomes. Therefore, before starting any expression experiment, it's necessary to start the analysis of previous results that have been published related to the same protein, or if it's not the case, to proteins that are similar, the same family, or proteins that are described for other species. Also, it's important to analyze the physicochemical characteristics of the protein that needs to be produced using bioinformatic tools. Also, there are several tools when designing the gene in order to obtain a DNA sequence which can be effectively transcribed and translated by the expression system. Also, there are several tools that need to be selected from strains, cell lines, plasmids, and promoters, which can give characteristics that can be important for the success of the process. Finally, it's important to take into consideration what are going to be the cultivation conditions that are going to take place in the process in order to obtain a screening of the best condition for the production of the protein. There is one factor which is really important when producing proteins in an heterologous expression system, which is the codon usage. Each expression system has a specific codon usage, and this codon usage leads to the system of the cell to produce proteins, taking into consideration that when a protein has several domains, in order to get a properly fold domain, it's necessary to have a reduction in the rate of translation in order to leave this first domain that is appearing enough time to get the proper conformation. Therefore, it's not a matter of having the highest prevalent codons in a specific position, but also to know which is the rate of translation in any of the domains that are present in a protein. So the result is that if introducing a recombinant gene with the original sequence in a prokaryotic expression system, as in this example, this will end up with the presence of several codons that are rare, that are not overrepresented in the expression system, and this might lead to a problem in the production of the recombinant proteins that in some cases can lead to aggregation or to proteolysis. Therefore, in most of the studies, this has been transformed into the modification of the sequence of the original DNA sequence of the heterologous gene into a gene that has in each of the codons a selection of the highest represented codon for the new expression system. However, this leads to a DNA that is not able to discriminate between regions that need to have a lower rate in translation, and in order to have this part of the protein properly folded. And I have selected one example of that in a recombinant gene that we have produced, that is the interferon gamma from bovine origin, that we codon optimized for E. coli and we tried to obtain in E. coli. The result of this experiment was what is shown here, in which we didn't get the full-length protein, we only get protein fragments. However, we performed another codon optimization, which was for the production of the protein in another prokaryote, which is Lactococcus lactis, which is a gram-positive. So in that case, when we produce this codon optimized gene in the Lactococcus lactis expression system, we obtain a nice protein band in the soluble cell fraction of the cell. And surprisingly, when we took this same codon optimized gene for Lactococcus lactis and we transfer that DNA, which gives rise to the same protein as this other gene, our surprise was that in that case, in E. coli, we could obtain this protein, this full-length protein, in the same expression system in which at the beginning we were not able to obtain this protein in a quality good enough for the purpose of the experiment. Another point that it's important to take into the consideration is the screening of cultivation condition of experimental conditions and the optimization of those experimental conditions. So first of all, several conditions need to be set under different ranges, and then those variables need to be designed in order to build a model, and after building a model it needs to be evaluated, and from the evaluation it's possible to obtain the optimization. So here I have an example of that, in which several variables have been analyzed in a specific ranges in order to obtain the optimum quality per ml of the recombinant protein that has been used in this experiment. So the optimal conditions for this type of experiment would be to use 40 grams per liter of glucose, and the temperature of the experiment would be 30 degrees. In any case, when trying to produce a recombinant protein, it's important to take into consideration the prokaryotic expression system E. coli, because it's the most used organism, and there are a lot of tools that are available, and also it's a system that is fast, and it's easy, and it's cheap. Therefore, the rule of thumb for this expression system is attempt that in E. coli. However, there are some exemptions that apply, as in the case of full-length eukaryotic proteins or integral membrane proteins. As I said at the beginning, aggregation it's a problem that it's present in most of the experiments when producing a recombinant protein. Therefore, when the protein is in the form of inclusion body or aggresomes, this cannot be used for most of the research and industrial applications. So there are several strategies that can be performed in order to improve protein solubility, as for instance to reduce the temperature in the cultivation, because then the rate of translation is reduced, and this is one of the points that can lead to protein to be aggregated. Also the use of weak promoters or partial induction, the optimization of the media composition, suitable cultivation strategy, and also a combination of that. So it's important to establish strategies to improve the protein solubility. Most of them are involving the reduction in the rate of translation of the mRNA that is being produced in the cell. There are some mutant strains that are able to cope with this type of problems, as for instance for proteins that contain disulfide bonds, there are mutant strains that are able to get an oxidized environment in the cytosol of Escherichia coli. Also, there are strategies which deal with the coproduction of folding modulators, which seem to be a limiting step in the folding of the proteins that are produced. However, if none of the conditions that have been taken into consideration give a good result, the last option would be to redesign the recombinant gene. All these procedures lead to the transition from the inclusion body to the soluble cell fraction of the cell.